So block 3, structure of pollen and ovule seed development. This block 3 we have to discuss. So in this block 3, that is the seventh unit is there. That is the structure of the pollen and ovule. Right? So okay. Now we have to discuss that is the structure of pollen and ovules. Different types of ovules are there. We have to discuss one by one. So the pollen grains is represent the male portion of the reproductive process in flowers, and ovules are the female reproductive parts. That is, we know that. So pollen grains are the produced in anther of stamen. The pollen grains produce male gametes which are fused with the ovule female gametes. This process or phenomenon called as the fertilization. After fertilization, the process will develop into the seeds in ovules after meiosis divisions. Megaspores are formed and megaspores developed into female gametophyte, which is produced the female gametes. So structure of pollen grains, we have to see here that diagram. So that photo means diagram here. Pollen grain structure means that exine and intine is there and germ pore also present. So pollen grains are the microscopic structure that is varies in size and shapes. So some are there very tiny, while other are the egg-shaped or different structures are there. Although too small see individually, they can be seen by the naked eye when they are in the large quantities. So viewed through a microscope, a pollen grain hardly looks real and extremely durable body. It has a tough outer coating. This hard coat offers the great protection from the harsh outdoor environment. This is the important because inside this tough shell lays two cells are there. That is the tube cells, and which is eventually became the pollen tube, and another one is the generative cell, which is contain the that male sperm nuclei, for which is used in the fertilization. These are three main components of pollen grains. That is, the inside the grain is made up of the cytoplasm. This fluid medium houses the aforementioned living cells, keeping the moist and alive. The outer shell is made up of two layers. The inside layer is applied named that is the intine. It is composed partially cellulose, and the outer layer is known as exine. This is highly sophisticated and complex outer layer, which is compound that is the sporopollenin. So waterproof resistance to the deterioration and very stiff. This shell is basically one of nature most advanced polymers. It is ensure that the tender cell inside have a strong chance of survival. So in addition, After time of the exile has folds, creases and spikes rising from its surface. So like extra armor, these features add to the protective nature of this layer. They also play an important role in the mobility of the grains, making more likely that they will be sticking to the legs of insects as well as catch the wind. So this is the structure of the male gametes means pollen grains structure. So coming to the ovals, types of ovals and what is the structure of oval, we are to discuss the process of formation of megaspore from the megaspore mother cells by the meiotic divisions that is known as the megasporogenesis. So this process takes place in the ovule. The ovule is considered to be an integumented megasporangium. The ovule consists of the stalk and body. The stalk is called funicle. One end of the funicle is attached to the placenta and other end to the body of the ovule. The point of attachment of funicle with the body is called hilum. Sometimes funicle get the fused with the body of the ovule one side and form a rigid known as a raphe. The body of the ovule shows two ends. The basal end, often called the chalagal end. The upper end is called the micropylar end. The main body of the ovule is covered with the two or envelopes called integments. These leave an opening at the top of the ovule, that one we call as a micropyle. So the integument encloses the large parenchymatous tissues known as the nucellus. The residual part of nucellus in the mature seed is called perismer. In the center of the nucellus situated the female gametophyte known as embryo sac. So here there are the ovules are related to integments. There are the two conditions are there. That is the uni-integument, bi-integument, and some other parts also there. Aerial, carancule, and etegmic conditions also there. So we can see here. So ovule with a single integument, that is the sympetalis, gammapetalus, cotyledons are there, means a single integument, that one we call as a uni-integument. Bitegment means with a single, here two integments are there. So aerial, another one is the aerial. This is a collar-like outgrowth from the base of the ovule, from third integument. So this aerial is found in the leech and nutmeg, etc. Carancule is formed out of both outer pigment in the micropylar region. Carancule is common ovule in the euphorbiaceae. Example, castor. Etegment. So etegmic, some parasite like Loranthus viscum. These are the integments such called as a etegment, means there is no integment, that one we call as a etegment conditions are there. So types of ovules depending upon the shape and orientation of the Ovals of angiosomes. There are the different six classes of types are there, or classes or types of ovals are there. That is the one Arthrotrophus, Anatrophus, Hemitrophus, and Amphitrophus, Arsinotrophus. These are the six oval types in the angiosomes. So Arthrotrophus oval, that is the micropylal, chalajal, and funicule are the straight line. So this is the most primitive type of the oval. Arthrotrophus means that is the micropylal, chalajal, and funicule. There are the same line, straight line only. Anatropus. Second one is the anatropus. The body of the oval is completely inverted. That is the 180 angle. They may be turned at the 180 angle. So the micropylar hilum come to place very close each other. That is the anatropus. Hemianatropous will turn at the 90 angle, 90 degrees up to the funicule of the body. So here the right angle to the funicule, the body of the ovule looks at the right angle to the funicule. That is the hemianatropous. So Campylotroph, so will is the circle and more or less at right angle to the funicule. Micropylal end to bend to downside, and that may be in the cruciferous members. And Amphitrophus ovule. So curvature of ovule is more, and embryo sac became the curve-like structure. That is the arch shoe type embryo is formed. So that is the Amphitrophus ovule and Sarcinotrophus ovule. The ovule is initially arthropous, but they became the anatropous due to the that is the unilateral growth of the funicule. The growth continually, the ovule one again became the arthropous. As a result, funicule completely surround the body of the ovule. That is example opentia. So Sarcinotrophus ovule, which is the funicule to formed as a like another integument, the completely surround the body of the ovule. So next one is the formation of megaspores. So the ovule or the megasporangium develop as a small prototubes of placental tissue in the very young ovule is a single hypodermal cell is the differentiated as the archisporium cell. The archisporial cell may be directly function as the megaspore mother cell, may be divided into pericranial to the form outer partial cell and an inner sporangium cell. The sporogenous cell directly behaves as megaspore mother cell. The diploid megaspore mother cell enlarges in size and divides by the meiosis to form a linear tetrad. Occasionally, T-shaped or inverted T-shaped tetrads are formed. So, megaspore is the first cell to form the gametophyte. So next one is the coming to the pollen storage. So how the pollen will store and preserve the pollen is useful source of diverse alleles with the gene pool and maybe the effective propagule for the gene banks. The ease of pollen storage and shipment of the potential for its immediate use provide researchers with the increased opportunity when they design their breeding techniques or to produce the new varieties. So method for pollen collection and desiccation, viability testing and longevity assessment has been developed from many species in the interest and have revealed the critical importance for the increased longevity um, by using high quality pollen desiccation. It's sufficiently in a rapid manner and subsequently storing it at very the low temperature. So reliable viability assessments are the dependent upon the adequate rehydration and the use of reliable strains. In vitro germination assess or in vivo pollination experiments, pollen preservation in gene banks will likely be implemented as a standard procedure for handling and assessing the development. So advantages to the use of pollen. The gene banked pollen can be made availability to breeder upon the require. For the tree species, this is the obviates the need for the growing the male parent in the breeding orchids. It allow the for wide hybridization across the season and geographical limitations. That reduces the coordination requirement to synchronize flowers and pollen availability for the use of crossing. With adequate pollen viability, so one can also load additional pollen into a stigma to increase the pollination and yield. The pollen available for the research programs as a single cell, the pollen provide a simple model system for research on the conservation and storage of pollen with the gene banks also ensure its availability year round for the basic biology and allergy. The research programs, the pollen captures diversity within the small, simple size and documentation is available for the long-term survival of the pollen from many diverse species. The pollen also serve as a source of genetic diversity in the collection where it is hard to maintain diversity with the seeds. Pollen can also shipped intentionally, often with the threat of disease transfer. So these are the some. So here these species are the liquid nitrogen at the low temperature. They are stored in the different terms. So next one is the pollen viability. So I show to the pollen viability. So this is the pollen viability germ tube. pollen tube growth. We can see here. Uh, so one commonly used vital stain is the fluorogenic ester and fluorogen diester. This is a test you measure the membrane integrity. Pollen grains fluorescence green when the cellular ester cleaves the FDA. So means fluorescence, fluorescent diestate. So since this assay is depends upon the functional membrane, the osmoticum of FDA stain. So is the often dissolved in the 100 for the 10 percentage to 20 percentage sucrose solution containing the boric acid and the calcium nitrate to the minimize the osmolysis and membrane leakage. So comparison of viability determined through the FDA or tetrazolium and those obtaining using the in vitro germination. In vivo tetrazolium tests reveal the consistently high correlation provided pollen in adequately rehydrated prior to testing FDA. So it is the correlation occasionally mm, been shown to give the false negative result where the viable pollen appeared dead. So several tetrazolium based stain are available for testing the pollen viability. So the many other vital stains have been developed and proper to put the around 50 years. The stains such as the Alexander and Estocaramine, Alanine, Blue and Xgal gel have been shown the successful identification of the viability test. So the viability result obtained with these stains may not correlate well with the in vitro germination assays. So the next unit that is the principles of hybrid seed production. How we are to produce the hybrid. So introduction, that is the eighth unit introduction we have to see. So hybrid is the processed or produced by the crossing between the two genetically similar parents. The pollen from the male parent will pollinate and fertilize the set of seed in the female plant to produce F1 hybrid seeds. For the production of hybrid crossing between the two parent is important. The crossing process will result in the heterosis. In nature, to create a genetic viability and for its wider adaptations in the different environment condition. So flowering plants has adopted many mechanisms for cross pollinations. So cross pollination result in genetic heterogeneity and show wider adaptations. So flowering plants have evolved a number of devices to encourage cross-pollination. Hybridization steps. It is one of the most method to produce the new crop varieties in which two or more plants of the unlike genetical conditions. So here the following steps are there. Selection of parent, selfing of parent, self-pollination crops, cross-pollinated crops. These emasculation and different methods also there. So we have to discuss one by one. So first, that is the step is the selection of parents. The selection of parents should be done with the great care, taking the into the consideration the objectives, so that the chance of selecting the desirable genotypes should be more from the segregating the population. As far as the possible of the potential type would be more from the segregating the population. So because they have the best adaptable while the selecting the parent, all the important characters to combined must be kept in the mind. So selfing the parents, this is the second step and is essential for the elimination of undesirable characters and also for obtaining the homozygous individuals. Selfing need not to be done it in the self-pollinated crops, while the cross-pollinated crops only have to be performed artificially. Self-pollinated crops. So artificial selfing is not necessary as they are the actually self-fertilized. So cross-pollinated crops. So inflorescence should be enclosed in a bag before anthesis to avoid cross-pollination. In crops like castor, whole inflorescence enclosed in the bag and maize selfing done by the collecting the pollen from the mm, tassels and dusting on the silk of the same plants. So emasculation. So this is the very important step in the hybridization. Emasculation is the remove the male organ in bisexual flower before the anthers and shed the pollen on the stigma to prevent the self pollination. So forced open method. In this method, the flowers are opened by the force before it is opened the naturally, and the anthers are removed with the forceps. This technique is useful to large size flowers like tomato, brinjal, chilies. Clipping method. This technique is usually used to cereals like rice, wheat. In this method, up to 1 by 3rd portion of the spikelets are cut with the scissors and anthers are removed with the forceps. So, this one is the very best method to. So, ring cut method. So ring cut method means a circular cut is made based on the corolla 2 mm away from the 2 millimeter away from the ovary. The corolla is removed and the anther is picked out the help of the force. So this method is generally used bendy, cotton and leguminous crops. So keel rupturing method. Papilionaceae flowers such as the black gram, green gram, and other some means red grams. No, so keel petal is ruptured and the stamens are removed with help of the forceps. Another treatment, hot water treatment also. So hot water treatment means remove the stamen with the forceps is time taking and in the plants having the small size flowers such as rice, jawar, ragi. So these caches is dipped in the mm, panicles in hot water for a definitive period time. At the so desired temperature is effective. This makes the pollen grains inviable without affecting the stigma and ovary. The panicle can be dipped in the thermoflask containing hot water. The temperature of water and the time of dipping differ from crop to crop and variety to variety. In rice, 45 to 52 degrees for 10 to 20 minutes. Cold water treatment. So this treating the panicle with the cold water is temperature around 4 degrees. That particular time make the pollen inviable. Another method is the chilling method is also. So chilling the plants and the particular length of the time to allow the temperature to make the pollen sterile. So in wheat, chilling temperature treatment here 13 to 14 hours at to 27 to 30 degrees to make the pollen sterile. So hot air method also there, chemical method, alcohol treatment, and male sterility method, self incompatibility. Another method is important, bagging method. So selected male and female flowers have been bagged separately to prevent the that is the contaminating the staminated flowers and cross pollination in the pollinated flowers. So bagging has been done irrevariability and processing even the day of crossing. Different types of bags used for this process. It is also essential to puncture the proper bags with the minute hole to prevent the mold formation, also provide the ventilation for the dispersing the seeds. Crossing. Next one is the crossing. These desired pollen anther have been collected from the male parent or either the petri dish on the paper bags. The collected pollen has to be brushed or dusted on the stigmatic surface of the emasculated flower. After crossing, the flowers are again bagged. So tagging and labeling and process have been properly bagged and labeled date and emasculated and number of flowers. So harvesting method. So crossed seeds F1 has been harvested after maturity and preserved after drying to safe mature level to raise the F1 generation. So crossing techniques are there. That is the and tagging techniques. So tagging techniques means this special technique is flowered in case of mango. The male and female trees are grown inside by side. The female parent is self-incompatibility at the time of flowering. Both the trees are enclosed in the insect-proof cage or net. So flesh is read hours of files and related inside. There is a cage is with the fertilizer facilities, cross pollination. Stones set on the female trees are collected to raise the F1 seedings. So next one is the modification of sex. So modification of sex, that is also important. So I shown to another diagram. So 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 the complete flower, the containing all four holes are there. That is the sepals, petals, androecium, and gynoecium. So incomplete flower, the flower missing any of the one hole. So imperfect flowers, the sexual distinctness, that is the monoecious or the dioecious. So perfect flowers, the gamete, that is the maturation in the different time, that is the dichogamy, that is the producer. This unisexual or infective love naturally leads, that is the outcrossing avoids the deleterious effect of inbreeding, depression, promotes heterozygosity, genetic viability, and genetic exchange, advantages to long-term survival and adaptations of the species. So monoecious, so flowers are unisexual or the present in the at the different position on the some plants. Example, cucumber. The terminal flowers are male flowers. In the middle of the plant is female flowers. Uh, the dioecious, the male flowers and female flowers are the different plants. That is called as a male plant or female plants. So sex modification through the hormones and chemical changes. What here? So in the chemical changes or either hormonal changes are there. That is the sex expression in the dioecious and monoecious plants and genetically determined and can be modified to the considerable extent by the environmental and introduced factors such as the mineral and nutrition, photoperiod, temperature, phytohormones. These are the so phytohormones have been that is the found to be most effect against for the sex modification and their role in the regulations of sex expression in the flowers plants. So the morphological difference in various sex types and their specific metabolic characters result from the uh, position of specific patterns of proteins, enzymes, and other molecules. So these modification of sex expressions in the cucurbids have been uh, introduced both the changing the environment condition and applying the treatment with the growth regulators. Agnes treatment to increase the female sex tendency while the gibberellins cause the shift towards the maleness. So that is the hormones and chemical inducing the femaleness. Auxins, that is the naphthalestic acid, ethanol, and ethophen, cytokinin, and brassinosteroids. So these are the to introduce the or induce the femaleness. So what are the chemicals used with the female maleness? Gibberellic acid 3 and silver nitrate ABA and thiopuranic acid. Another one is the Talmin and paclobutrazole. These are the two increased or induced the maleness. So environmental sex modification. So environment has a greater influence on the sex modification, but due to the introduction of the photosensitivity varieties are hybrid in the modern era of the agriculture, it has gained less importance. However, in the seed production, it has its influence on the sex expression in the cucumber and High temperature and long day length, that is the favor the male flowers. So high temperature will extend the flower of male flowers. As the temperature increased from 19 to 25 degrees, the mode of the first male flowers has also increased from 9 to 16 in number. This is the clearly indicates that the high temperature favor the male and delay the female flowering. So male sex expressions of several plant species flower favored by the high temperature and female sex expression by the low temperature. So male sterility match mutant of tomato developing the male sterile flowers at the minimum temperature of 30 degrees and normal flowers at low temperature. In Brussels sprouts of low temperature effect on the developing the androecium. In onion, in the slight production of viable pollen by the normal male sterile plants that is above 20 degrees. So next one is the seed development and heterosis. That is the eighth unit is there. Means sorry, ninth unit, third unit, third block, ninth unit. That is the seed development and heterosis. So seed development and heterosis here to discuss. That is the ninth unit. So after fertilization, the development of finalized ovule into the mature seed ovules several different stages. Seed formation begins with the embryo sac with the certain expectations with which is the about the same in shape, size, and arrangements. The integument of the ovule becomes seed coat of the mature seed. Normally, the nucellus is observed. The nucellus may persistent in some such general, that is the cloves, rubber, papaya, pepper, beetroot, etc. So thin layer of called perisperm laying the inside the seed coat, that is the natural material for the embryo. So endosperm serve as the principal nutrient support for the embryo of many species. Monocotyledon, that is the embryo endosperm, usually reach the maximum morphological development at physical maturity and persistent to comprise a major part of the seed. In dicotyledon species, the endosperm may not develop or may be used by the using the developing embryo or comprise known as a small part of the seed. That is the two cells and undergoes into the transverse division forming the two upper imposed cell. These result T-shaped four-cell proembryo. The cells are divided transversely and forming the suspensor. Cell and its derivatives undergo vertical division and forming the group of 4 to 6 cells. This group divided by the ubiquitous root cap. So the two cell form the result of the division again divide vertically forming the quadrate. So each of the quadrate divide transversely and octant containing the two types of the cells are formed. The cells of these octant undergo vertical division resulting in the globular proembryo. Periclinal division occur in the peripheral cells of the globular proembryo that delimit an outer layer and determine tangent. The tier 1 gives rise to the cotyledons and shoot apex from the hypocotyl radical axis. The certain deviations from the above parent and embryo development are found in the different plants. So the different types of embryogeny are distinguished depending on the plane of the division of the apical and extent of the contributants of the basal cell towards the embryo development. In monocotyledons, the cell remain undivided and develop into the ostoria of the suspension. The cell divides into transverse divisions and gives rise to uh, two caps. The terminal cell develop in the grasses in the different from the other monocotyledon. In the dorsiventral symmetry is established as a result in the peculiar oblique positions cells and early in the embryogeny. The single cotyledon is reduced to observed scutellum and additional structure like coleoptile and coleorhiza are formed. Endosperm development, these are the three types of endosperm development, that is the nuclear, cellular and hilobile endosperm, where the endosperm nucleus undergoes the several divisions superior to the wall formation. Cellular endosperm, which is the no free nuclear phase, and hilobile, where the free nuclear division processed and followed by the cellulization and accumulated in the endosperm from adjacent tissues. In endospermic dichord endosperm they retained as a permanent storage tissue. In non-endosperm dichord endosperm reserves are depicted and calculated by the developing embryo. So, as a part of endosperm in the uh, depleted in the cells during the embryo nutrition, and this lays as a layer between the starchy endosperm and scutellum. So seed coat integuments of the ovule undergo marked recognition and histological changes during the maturation to form the seed coat. Right? So here, so the seed growth when the soybean represent the monocots and dicots may illustrate the changes in the pattern of the accumulation of reserve material in the different stages. So next one is the hybrid seed production. So see that hybrid seed production, you can see. So ultimate, the aim of hybrid seed production is to produce the quality seed. The following four steps are followed. General hybrid seed produce the choice and development of seed parent, choice and development of restore or male parent, and maintenance and multiplication of parental seed, production and improvement of F1 hybrids. So these four steps when we produce the hybrid plant. So see here, that is the diagram. This one is the hybrid plant to produce how we have to discuss. So choice and development of seed parent, that is a seed parent or the female parent of the commercial hybrid should be male sterile line. But in case of non-viability, suitable male sterile line, the fertile line or self-incompatibility line can be used where the manual labor will be needed. So in the case of food grains crop, where the number of seeds produced in such pollination is single pollination will be produced many seeds. There are fertile pure lines. So in case of the monocotyledons, the plant like maize, the male flowers can be removed easily. Those are the born on a species, a piece of the plants. After identification of the male sterile lines, it should be maintain the isogenic B line. So strains can be identified to have B line, which is reaction by the crossing them to A line. Then the potential B line is converted to A line by the process of repeated back crossing till the date. So transfer of male sterility is required when the seed susceptibility or unwanted agronic stain are associated with the male sterile characters. This can be done two ways are there. That is a single phase repeated back crossing and two phase limited back crossing. So next one is the heterosis. What is the heterosis? Means here the heterosis has been known since the art of the hybridization came into the existence. So um, Kohler was the first to report the hybrid vigorous in the hybrid tobacco and datura. So when the two homozygous inbreeds, they are obtained by the continuous inbreeding of genetically unlike constant, the cross together and resulting by the hybrid obtaining from the crossed seeds are usually robust, vigorous, productivity and taller than the either parents. So this increased productivity or superiority over the parents known as the heterosis or hybrid vigor. Heterosis can be defined as the superiority of one hybrid over the both the parents in the terms of yield or some other character. Right? So, so this one is the for the so nice introduction to produce the structure and structure of the pollen and ovules and seed development and pollen viability and other one is the hybridization, seed hybridization, heterosis. We have to discuss, right? So thank you.